Introduction to Biotechnology, the Basics of Biosciences. Biotechnology is a fusion of biology and technology, leading to innovations that span across medical, agricultural, and environmental sciences. This foundational lesson aims to introduce you to the core aspects of biotechnology, explaining complex biological systems in an engaging, story-like manner, and illustrating how these systems are harnessed to develop technological solutions. 1. What is biotechnology? Biotechnology involves using living organisms or their products to develop or make modifications to products or processes for specific use. It encompasses a wide range of procedures for modifying living organisms according to human purposes. Example 1. Production of biopharmaceuticals like insulin, which uses genetically modified bacteria to produce human insulin. Fermentation processes in making beer and yogurt, employing microorganisms to convert sugars into alcohol or lactic acid. Example 3. Bioremediation, where bacteria or plants are used to clean up contaminated sites, including oil spills. Example 4. Genetic modification of crops to enhance yield, nutritional value, or resistance to pests and diseases. Historical perspectives in biotechnology. While the term biotechnology is relatively new, the concept dates back to ancient civilizations. Example 1. Egyptians using yeast fermentation to brew beer and bake bread around 5000 BCE. Example 2. The selective breeding of animals, like dogs and horses, by ancient civilizations to enhance desirable traits. Example 3. The use of moldy soybean curds as an antibiotic in ancient China. Example 4. The domestication of plants, like corn and wheat, which were genetically modified through selective breeding by early farmers. Draft 3. The building blocks of life. Understanding cells, the basic units of life, is essential in biotechnology, as many technologies are designed to interact with or manipulate cellular processes. Stem cell therapy, where cells are used to regenerate damaged tissues. Example 2. Cancer research employing cellular cultures to study tumor growth and test potential treatments. Example 3. The use of CRISPR technology to edit genes within human cells in a targeted manner. Example 4. In vitro fertilization, IVF, a process that involves manipulating sperm and egg cells to help with fertility. 4. DNA, the blueprint of life. DNA carries the genetic instructions necessary for the development, functioning, and reproduction of all known organisms and many viruses. Example 1. Genetic testing, which can reveal genetic disorders or the likelihood of developing certain diseases. Example 2. Forensic analysis using DNA fingerprinting to identify individuals or determine familial relationships. Example 3. The Human Genome Project, which mapped the human DNA sequence and opened up avenues for gene-based therapy. Example 4. Genetically modified organisms, GMOs, where DNA is modified to express desirable traits such as drought resistance in plants. Proteins. The workhorses of the cell. Proteins perform a vast array of functions within organisms, including catalyzing metabolic reactions, DNA replication, and transporting molecules from one location to another. Example 1. Enzyme replacement therapy, which treats diseases by providing patients with essential proteins their bodies cannot produce. Example 2. The development of synthetic hormones like growth hormone, used to treat various human growth disorders. Example 3. Antibodies, protein components of the immune system, engineered to target and neutralize pathogens. Example 4. Biocatalysts, proteins designed to speed up chemical processes in industrial applications. 6. Enzymes, nature's catalysts. Enzymes are proteins that act as catalysts to accelerate chemical reactions, crucial in both natural processes and industrial applications. Example 1. Lactase added to milk to break down lactose, making it digestible for lactose-intolerant individuals. Example 2. Pectinases, used in the clarification of fruit juices by breaking down pectin, a structural polysaccharide in plant cell walls. Example 3. Industrial enzymes used in the production of biofuels from plant biomass. Example 4. DNA polymerase, an enzyme used in PCR to amplify DNA sequences, critical in genetic research and forensic science. 7. Biotechnology in medicine. Biotechnology has brought about significant advancements in medicine, enhancing diagnostics, treatment, and patient care. Example 1. targeted cancer therapies that specifically attack cancer cells without harming normal cells. Example 2. Monoclonal antibodies engineered for use in immunotherapy to treat diseases such as arthritis and cancer. Tissue engineering, including the development of artificial organs and tissues for transplantation. Example 4. Genetic testing kits that allow for the early detection of diseases like breast cancer and cardiovascular disorders. 8. Biotechnology in agriculture. Biotechnology has revolutionized agriculture, enabling the development of crops that are more nutritious, require fewer chemical inputs, and are more resistant to stress factors, such as pests, disease, and variable climate conditions. Example 1. Bt crops, such as Bt corn, which are genetically modified to express a bacterial protein that is toxic to certain pests. Example 2. Golden rice, genetically engineered to produce beta-carotene, a precursor of vitamin A, to combat vitamin A deficiency in the developing countries. Example 3. Drought-resistant crops engineered to withstand periods of low water availability, enhancing food security in arid regions. Example 4. Herbicide-resistant crops that allow farmers to control weeds without harming the crops themselves. 9. Ethical considerations in biotechnology. As biotechnology touches on manipulating life, it raises significant ethical and social questions that require careful consideration. Example 1. The debate over GMO safety and labeling, focusing on consumer rights and environmental impact. Example 2. The ethics of cloning, particularly concerning cloning animals for agriculture or endangered species conservation. Example 3. Considerations around the accessibility and affordability of advanced biotechnological treatments in medicine. Example 4. The implications of gene editing technologies such as CRISPR and their potential use in human embryos. 10. The future of biotechnology. The future of biotechnology is ripe with opportunities for further innovation. Emerging technologies and applications are likely to transform current practices in medicine, agriculture, and environmental management. Example 1. Synthetic biology, which designs and constructs new biological parts, devices, and systems, or redesigns systems that are already found in nature. Example 2. The use of nanotechnology in biomedicine for targeted drug delivery systems that could improve the efficacy and reduce side effects of treatments. Example 3. The development of bioartificial organs, which may one day replace the need for donor organs in transplantation. Example 4. The potential for biotech solutions in combating climate change, such as carbon capture and storage technologies, or biofuels that reduce reliance on fossil fuels. Conclusion. Biotechnology is a dynamic field that combines biological sciences with technological innovations to solve some of the most pressing challenges facing humanity. From improving health outcomes and agricultural efficiency to addressing environmental issues, the potential of biotechnology is vast. This introduction has aimed to provide a comprehensive overview of the field, making complex concepts accessible and engaging for beginners. As we progress through this series, each lesson will delve deeper into these topics, exploring the science, technology, and ethical considerations that drive the biotechnology industry today.